My name is Urmi. I am a postdoctoral research fellow at Harvard Medical School in Boston, Massachusetts. I work, my research is in biochemistry and genetics, and my PhD, which I got last year, is in biochemistry. So I was invited here today to talk to you about, I understand a lot of you are interested in biomedical careers, be it research, medical school, etc. So I'll try to tell you how I got to this point, right from college to graduate school and then now at Harvard. I put together a few slides and feel free to, I think there will be a question session after this, but feel free to ask questions even during my talk. I think you can type in your questions. So, my name is Urmi. Like I said, my PhD is in biochemistry. I got that, I graduated May 2019. Right now, I am a postdoctoral researcher at Harvard, which means that I have run my own independent research at Harvard Medical School. I am really interested in, I love teaching and mentoring undergraduates, and I've worked extensively with undergraduates throughout my career. I thought I'll put together a little timeline about what a typical research career looks like. So I got my bachelor's in chemistry, and that was in 2007. So I went to a program that had a 3-year chemistry course. And I think I love chemistry. I think it's a very it's an amazing subject to learn if you want to get a good foundation of the biomedical sciences. After that, I transitioned into biology. I did a Master of Science in Biophysics and Molecular Biology. That was in 2012. After that, I took a gap year, and in this gap year, I had 2 roles. And let me tell you a bit about the schools I went to for my bachelor's and my master's. So for my bachelor's, I was born and raised in India. So for my bachelor's and master's, I went to schools in India. So I went to St. Xavier's College in India, which is one of the top schools in the country. And I majored in chemistry and minored in mathematics and microbiology. After that, like I said, I got a Master of Science, which is like a 2-year course. And I got that in University of Calcutta. And I studied biophysics and molecular biology. And this was the period I really transitioned from chemistry into biology. And so it was like an amazing learning curve for me. Next was my gap year, like I was saying. And I think my gap year was really interesting because I had a dual role. So I worked as a lecturer with graduate nursing students. So I was teaching at the graduate nursing school. And I was like, I think 22 at that time, and all my students, they were already nurses. So this was like an advanced degree. So they were all almost double my age because they were like professionals in their field. So this was like a very, I was overwhelmed in the beginning, but it was like an amazing experience. And these nurses were so professional. And that kind of taught me a lot of things about how to handle yourself in a professional environment. So I worked as a lecturer for this 1 year, and I also, at the same time, I was a clinical research assistant, meaning I worked on cervical cancer. As you know, it's this cervical cancer is one of the leading causes of death due to cancer among women, and I worked on technologies to detect cervical cancer. So in developing nations and even in rural areas, cervical cancer detection is a huge problem because there aren't that many resources and there are simply too many people to screen. So there is active research going on which aims to figure out how we can screen how we can come up with new methods which are more economic, which are more robust, and which can screen more women in a shorter period of time. So I was involved. I was a clinical research assistant, and we sought to work with. We sought to address these problems. This was a very, I should say, significant period in my career because I simultaneously was involved in medical education and medical research as two parts of my job. And I understood the importance of how research can really touch people's lives. And you can do that when you really work with patients. We work with patient samples. So I worked at an institute where there was the hospital and the research wings, and we would go into the hospitals to get our samples from patients, and we interacted with patients, and it was really, it kind of made a huge impression about how your research can really impact people. So in the next phase, so this was my gap year, and then in 2013 I moved to the US and I came to graduate school, and my PhD was in biochemistry. So I came to Rutgers, which is the State University of New Jersey, and it's really very beautiful, as you can see. And here I worked in the Rutgers School of Public Health, and I studied my thesis was, my doctoral work was, the title of my thesis was Mechanism and Regulation of Yeast in Human Mitochondrial DNA Transcription. So that sounds a mouthful. What that is about is, what I studied is basically, you guys must have read in school about the central dogma. And let me just restate what that is. Central dogma is really the flow of information, flow of genetic information from DNA to RNA to protein. As you know, in higher eukaryotic organisms and even in most prokaryotes, our genetic information is stored in molecules called DNA. DNA gets converted to RNA. DNA is, I shouldn't say converted, RNA is made from DNA through a process of transcription. And then we have proteins made from RNA through a process of translation. So what I studied is really this process of making RNA, which is called transcription. Now, I specifically looked into mitochondria, and as you know, mitochondria is the powerhouse of cell, and how mitochondria was formed. And this is like a really interesting cartoon that I see. So there was this huge prokaryotic host cell. So this is prokaryotic, meaning it's very rudimentary, it's very ancient cell. And this guy, while roaming around, came across this alpha proteobacterium, which is essentially a kind of bacteria, a smaller bacteria. And what this big prokaryotic cell does is simply eat up this tiny bacteria. Once this event happened, so this was like a long time ago. It was 2.7 billion years ago that this event happened. Once this event happened, we have this a cell which has its own components, and it has all the components of this smaller bacteria. Through evolution, this eventually became a eukaryotic cell, which is where we come from. We are eukaryotes. And what this has is, it has its own set of genome, it has its own DNA, and it also has the DNA of the tiny bacteria that it ate up. This compartment eventually became what is now our mitochondria. So mitochondria is like its own world. It's like it's kind of like an autonomous country. It runs itself. It can have its own replication, which is the process of making DNA. It has its own process of making RNA. It has its process of making its protein. So mitochondrial, so right now through evolution, what this looks like now is mitochondria has, so right now what your cell has, eukaryotes, we have two sets of genome, which is something we don't really think about. Because whenever you think of your DNA, you think, oh, it's in the nucleus. Yes, that is correct. Most of your DNA is in your nucleus, but there is a small portion of your DNA that is in your mitochondria. And this is what I focused on. I looked at the genome in the tiny compartment that you usually do not think about. Mitochondrial transcription machinery is a completely separate transcription machinery that's completely separate from the nuclear machinery. It consists of a huge protein called polyRNT, which is the main polymerase. And this is the guy that makes the RNA. This will add your ribonucleotides one after the other and make a long RNA chain. However, polyRMT or the polymerase is not independent. It has to be assisted by smaller proteins, which is shown in gray and blue. And these blue and gray proteins, these two factors, help this polymerase make the RNA. And how, when it would, it's a huge mystery when these two assistants come together to help the main polymerase, when they refrain from doing so. And that's how regulation in the mitochondria happens. Now, if you remember, in your body, energy is made in the mitochondria. That is the only source of energy in your cells. So how you breathe, how your heart works, how your kidneys function, everything is dependent on your mitochondria. So if your mitochondria stops producing energy, you won't be even able to blink your eyes. So that's how important this is. So how transcription happens directly controls how energy is made. And so this process is, mitochondria is critical in health and disease. It's, as you know, present in all eukaryotes, and it produces this energy currency, which we call ATP. This is like money. You spend money and then you say buy something from the store. Similarly, you use this ATP, you use this energy, you give this energy to all the organs in your body, like heart, like eye, like liver, kidneys. All these organs get their energy from mitochondria. And if you stop making that energy, you can have a wide range of disease, right from heart attacks to brain seizures and diabetes, etc. So that's why that's why we study this process. There is a huge, I think under Barack Obama, there was a huge fund, NIH, the National Institute of Health, started the Brain Heart Initiative, which poured in a lot of money into mitochondrial research, and is one of the most exciting fields in biology. So that's what I did for my PhD. And my research was supported by various sources. So I had a very successful funding source. I was able to get my own sources of funding, and they were from Rutgers. They supported the first and the last year of my doctoral studies. So PhDs typically for me was like 6 years. So year 1 and year 6 were supported by these two fellowships from Rutgers. And year 3 and 4 and 5 were supported by the American Heart Association. American Heart Association funded so I kind of wrote a fellowship and sent it to them, and they funded my project because it's critically important to their mission. And my research support was also given by the National Institute of Health. This is how my lab was funded. My advisor had funding from the National Institute of Health, from the Basic Sciences Division, and all these three sources helped support my research and my fellowship. So I also was awarded the the top dissertation for my thesis, and I was and then I decided to I had a wonderful time in graduate school, and then I graduated in 2019, and I wanted to keep on studying transcription because I just think it's just a very fundamental process to study if you want to really understand gene expression programs. So I decided to move to Harvard for my postdoctoral research, and this is where I work now. This is the iconic quadrangle from Harvard Medical School, and here what I study is I went into nuclear transcription, which means that I decided to focus on the main genome of the cell, which is in the nucleus. Like, you know, there are string-like particles inside the nucleus that constitute your DNA. These ribbons or strings, like you would say, have a sequence of information that we call DNA. And this DNA is, the transcription is carried out by another kind of polymerase, a different polymerase from the mitochondria, which is known as RNA polymerase 2. And when this RNA polymerase 2 makes the RNA, it also adds a modification to the 5' end of the RNA, which is commonly called capping. Many of you might know this, that there are several modifications added to the RNA. So these modifications are really like stamps that you attach to your letter. So if you want to post a letter to someone in, say, Massachusetts, you have to put a stamp on your letter. And that's how you know, that's how the post office knows that where this letter has to go. RNA is the same thing. There are millions of RNA molecules that are being made in the cell. So how would the postal service of your cell know where to send the RNA? So what they do is they kind of code the RNA. So they kind of add specific modifications to the RNA. Okay, you need to go to Massachusetts. So let's like add a red modification to these 10 molecules of RNA. You need to go to Texas. So let's mark you differently. So that's what RNA modification is. The different kinds of modifications kind of tell the cell what to do with this RNA. So I specifically study some sort of one modification that is done at the 5' end of the RNA, and this is called capping or G capping. And there are specific enzymes that cap this RNA, and this decides where your RNA will go, how stable your RNA will be, what functions your RNA will do. So right now I study a very specific topic in transcription, which is the function of capping enzymes in transcription. How do they regulate transcription? How would they regulate transcription if you were to get some form of cancer? How would they regulate transcription? What is happening to this transcription when you're getting a heart attack, normal versus in a cardiac arrest situation? So that's the kind of thing I study right now. So that was a bit of my journey, but I did not. So here, this is a picture of me standing on top of the Empire State Building alone. But trust me, I didn't get there alone, and there were so many people, my mentors, that were involved where I am now. First of all, are of course my parents, who have always been very supportive of everything I've done. Here is a picture of me at my convocation, at my graduation. These were my lab mates. These were my peers, and they're amazing people. My graduate advisor, who was amazing, who is an amazing scientist and really an inspiration, and my husband, who is also a scientist and has helped me extensively throughout my graduate career. So what I do right now is I live in Cambridge actually. Here is a picture of the Charles River between Boston and Cambridge. It's really beautiful. And here is Harvard Medical School. We can never take enough pictures of Harvard Medical School because it's just so pretty and it's just beautiful. And this is the Massachusetts State House. So I haven't seen a lot of Boston, but I'm very excited to be at Harvard and be a part of this community in the Boston Cambridge area that is so intellectually vibrant and diverse and very accepting, innovative. It's I think it's heaven for a researcher to be here, and it's really a privilege. So that is a little bit about my journey so far. And let's we can go into questions. Why did you decide to pursue a career in your specific field? So I didn't have one moment where I decided that. That didn't happen. I was consistently extremely interested in chemistry, and as a kid, I would like add different things and do reactions in like whatever, in a glass or in a cup or anything that I could get hold of. And it's a miracle that nobody was injured during the process, because I'm sure some of these reactions should not be done unsupervised. But I always loved chemistry, and I think uh, it just I had friends who were also interested in chemistry, and we were part of like clubs and stuff. So as I grew older, I just loved it even more, and it was kind of consistently built up. And finally, I decided that this is what I had to do. So there wasn't one moment.
Would you suggest to anyone else who had a passion for something as strongly as you did for chemistry to try to pursue this into, college and beyond as well?
I would definitely recommend that, and I think it's also I would highly recommend trying it out before you actually choose that career. Like do a summer internship or shadow someone to really figure out if this is for you. Because research is not easy. Research is pretty hard, and you have to be very consistent. You have to be very like you have to persevere, because some of the problems because most of the problems you don't ask easy questions as scientists, otherwise we won't be solving anything. So I would definitely recommend uh, uh, talking to people, talking to more scientists, and figuring out if this is for you.
How has doing research changed while in quarantine during COVID?
Oh, that's a very good question. It is. It has been. So I joined. I moved to Boston in December 2019, and it has been pretty rough as a researcher because our labs completely closed down. Harvard kind of following city mandates completely closed down our research facilities. So we were able to get nothing done. We could not go into lab. We were not authorized to go into lab. So it was like zero productivity for several months. And then everything start. And remember, in this time we have some lab. We don't have cell lines, but many labs have cell lines, and these have to be kept uh, maintained all the time. And it was extremely stressful doing that amidst the lockdown. So my research, and I'm sure, pretty sure, a majority of scientists will agree with me that my research has been severely hampered because of COVID.
Do you think something like this, a pandemic, could negatively affect society in a way that we can't really use a lot of the research that we could be doing?
I think there is substantial loss in research productivity because of the pandemic. But I think that especially the young generation, people like you, should use this as a motivation to become scientists, to become doctors, to become healthcare workers, because it is you have the knowledge, and you will have the knowledge, and you will have the power if you choose to be a scientist or a healthcare professional to help people. So I think you always have to look at the uh, silver lining, and the silver lining here is that people realize how important it is to do research. I mean, I know a lot of people who don't think about research, and now they are suddenly, oh, we need a vaccine. I mean, how will you have a vaccine? You need to have clinical trials and basic research to get the vaccine. So I think this should be a huge wake up call for the younger generation to get motivated and become scientists. So you should use the silver lining.
Do you have any advice for high schoolers or college students who want to pursue research?
Yes, so I have worked extensively in a research setting with high school students and undergraduates. And I think my advice would be to definitely get as much experience, a shadowing experience, or having your own project, whatever, in whatever form it is, to get some like actually get your hands wet with research, like actually doing research rather than just taking a course about research. Because I think working in a lab setting, it will kind of tell you about yourself, like if you will thrive in such an environment, if research is really for you. And it's totally okay if you decide that research is not for you. You try it, you don't like it, that's totally okay. But I think you should be in a position to make an informed decision that this is definitely for you, and not because your friend is doing research and hence you need to do research. So it should not be like that. This is difficult, but you definitely can do it. So my advice would be to definitely talk to people, reach out for internship opportunities, etc., as in high school or as undergrads, and get that kind of feedback.
Have you faced any setbacks on your path in STEM, and how did you overcome them?
So when I was in college, I think I was like 18 then. In as a freshman, I got extremely sick. I was like bedridden for like 3 months. I think I had to be taken to the hospital. So I was going through a lot of health issues at that phase, and there was in that phase I thought that maybe I could not do a job as demanding as being a chemist. Uh, uh, in that situation. So that was the huge challenge that I faced. But again, I had friends who helped me. My parents were very supportive, and I kind of fought back from that. So my advice would be to you just have to keep pushing. If you really want to do it, these hurdles will come your way. There will be several challenges that you will face, but you just have to keep at it, even if you're not getting the results that you want. it's okay, but you just have to keep going, just and never give up. It's really, that's what matters in the long run. You just have to try again and again.
Has there ever been a time where you regretted being a scientist and wanted to do something else?
Never.
I can definitely sense the passion that you have for this field, so that answer wasn't very surprising at all. How did you get your research position at Harvard, and do you have any tips for those who want to work in a lab with a professor?
So getting into Harvard is definitely like super competitive because everyone around the world wants to get in here. Like you have people from across the globe that want to do research at Harvard because it's just amazing. So I got my I had a pretty successful graduate career, and I had published well, and so I kind of got in touch with my advisor. I think I directly emailed him, and we kind of set up a interview, like meeting, and then I came to his lab to. I also wanted to assess whether this environment is right for me, because the environment is very important, because you spend so much time with your lab mates, and if you don't like them, it's not worth doing such a thing. So I so that's how I emailed him, and I talked to a lot of people, and I heard about his lab. Again, networking, talking to your peers is like key to thriving in science. So I got it through contacting my advisor, peer mentoring, talking to friends and other scientists. So that's how I got my position. And as far as if you want to work in a lab with a professor, yes. So I think my advice would be to do not write generic emails. That is a huge uh, uh, it's pretty, it looks very bad on your part. And people can tell if you have written the same email to 20 other people. People can actually tell. Trust me, people can tell. And I've read so many of those emails where they're writing about, oh, I want to do this in your lab. Well, the lab doesn't do this. Like, this is from somebody else's lab that you're writing. So don't do that. Do not write generic emails. So if you are interested in someone's lab, you don't have to write a lot of details, because at your stage you might not be able to understand everything they're doing, but you can overall give an idea about yourself, why you're interested in doing this, and write a very sincere email, and just say that, oh, maybe this is the area I want to work in. Show people that you're genuinely interested, that you're serious about this. We have also had many people in our lab, like undergraduates, who get into the lab and never show up. So don't be very professional, because everybody else will be professional with you. So that is what is expected of you. So that's why it's very important to be honest. Like when you go in the lab, tell them ask them what their expectation is. They might have a fixed number of hours, say, oh, as an undergraduate, you have to work, say, 12 hours in this lab. Make sure you have 12 hours on your schedule that you can spare in this lab, and you just don't show up for an hour or don't show up at all. Make sure, because it's like burning a bridge. It would just look very bad on you would look very unprofessional if you do that. So first thing is really assessing how much time you have, how much time you can invest in research, and committing to that. It's very important, because when you work in a lab, there will be other people training you. So your schedule is kind of tied to their schedule. So they're not going to be happy if you don't show up and don't do the experiment that you were supposed to do. Second thing, we use very expensive reagents in research, as you know. So if you are not 100% in the game, you will end up wasting resources, especially in biology and chemistry. Everything is so expensive. So they need to trust you. They need to know that you are here 100%. That saying, don't be afraid to make mistakes. Everybody makes mistakes, but learn from those mistakes. Don't make the same mistake again. Write things down, everything, even the simplest calculation. I till to this date, I've been in science for like 10 years, I think now, more than that. I always write everything down, because if something doesn't work, you can come back to your notebook and you can troubleshoot. Oh, I made a mistake here. And then you're fine. If you can catch a mistake, it's fine. So I think when you approach a lab for to do research, make sure you write a very customized email and very sincere email. And if you don't get a reply, don't get discouraged. Just follow it up in like 2 weeks or something. You can even try contacting somebody else other than the principal investigator in the lab in case you don't get a reply. Sometimes advisors are just too busy to look at emails. And then once you get in the lab, be very well. So they will, if they're interested, they'll call you for an interview or a Zoom session these days.
Mhm.
Be very honest about the time commitment that you are up to. Like, oh, I can spend say one day 8 hours in this lab, and they will give you projects accordingly. If you say I can only spend 8 hours in a week, that is completely okay. But just be honest about it, and have fun. It's a lot of fun. So you should never forget the fun part of being a scientist.
Like how do you join the researches that you were in? How did you conduct them? And in your case, how do you like start them and work on them after that? And if there is a certain college or place that you need to go to.
So I'm guessing this person's interest is the biomedical sciences. So you can have various tracks to go into a PhD or an MD PhD. Uh, I work with two undergraduates during graduate school. Both of them went to med school. They are in MD programs, and they did research with me. So I think one of them, they were both on the pre med track. So you can consider doing that. So there will be, if you meet your undergraduate advisor, they will tell you that these are the requirements of pre-med. There will be a set list of courses in biology, chemistry, etc., that you need to take. So you take those courses. So the key here again is to communicate very clearly to your undergraduate advisor, and they are very, very helpful when it comes to helping you design your curriculum. And this curriculum, of course, has to be tailor made to what you want. So you take those courses, and of course, you have to do very well in those courses because getting into graduate school or MD PhD medical school it's very competitive. So once you do that, try to get some research experience in during your undergraduate degree. As an undergrad, there will be several. You talk to your dad usually, like where I went to graduate school, we had this undergraduate coordinator kind of person, and she was responsible for helping undergraduates find labs. So identify that person early on in college. As a freshman, you should even when you just get into college, you should be very aggressive about finding a lab. So because it's very competitive getting into a lab, so everyone is looking for it already, so you better look for it pretty early. And then once you if you like the research, you can continue doing research. My undergraduates worked with me for 3.5 out of 4 years that they were in college. So that's like they started in their freshman year and they published with me, and that's really important. I think you should always try to publish your work. That's kind of distinguishes yourself. So that would be my advice. Just go get into a program and do all the things that is required for pre-med requirements, and do research to determine early that, okay, do I really want to do research? Do I really want to be a doctor or a scientist or get an MD PhD, whatever it is? And the only way of doing that, of understanding what you really want, is just doing research. So that would be my advice. Just get into a lab and just do some experiments.
If you had any advice or if it's possible to do a PhD quickly?
No, there is nothing called a quick PhD. If you think you'll get a quick PhD, you will not do a good job, or the quality of your research will not be very competitive. However, people do get lucky and they do make discoveries, but discovery of that proportion is very rare for a graduate student. Let me tell you that. Like, you see in movies that people suddenly find things and, I discovered this. That rarely happens, okay? Discoveries are made through decades of work. And if you look at Nobel laureates, you'll see that it's really years of work that build up to that. So if you it's PhD is not for you if you're trying something quick.
If you're saying that the limiting factor is discovery for that?
Limiting factor it is discovery, but at the same time, so you really want to, so so what is research? Research is really asking a question that nobody has ever asked before. So when you take exams in school, there is a right answer, right? Like you have to select the right answer, or you have to write down the right answer. In research, there is nothing called a right answer because nobody knows that answer. The question you're asking is so specific that only you can figure out that question. Like maybe people have asked this question before, but nobody has satisfactorily answered this question before. So there is so there are no right answers. So just setting up your system to getting all your reagents, making proteins, having cell lines, making knockout mice, getting patient samples, whatever kind of research that you do, it takes substantial amount of time to set up your system, understand your system, analyze your data, uh, look for false positives. Like something may seem like that, but you have to run several kinds of experiments to make sure that this is what it really is. So I think the limiting factor in research is really validation. You need to validate your results. You need to be confident about your results, because if you say, it's like the vaccine trials that we are having now. They have this drug that will give us immunization from COVID, but you need to know for sure, because this drug is going to be given to human beings. You better be sure that this drug works. You better be sure that nobody's going to die when you give that vaccine to people. And so that should make you realize that if you want to do it quickly, you won't do a good job. That's why we have all these standards, these regulations by FDA, that it has to go through these number of trials. We have to test it in these number of people, and that is not quick. That is very methodical. That is very detailed. And so I think the limiting factor is really validation.
If you could go into a bit more detail about how you were able to identify the resources each school you attended had, and how did you like reach out to your advisors and professors to make use of those resources?
I think one of the critical things in your career is identifying resources. When you are in college, you are one of, say, thousands of undergraduates if you go to a big school like Harvard has like thousands of undergraduates, you can be lost very easily. And that is a very common feeling in freshmen, like, how do I do this? And I think the first thing you do is identify resources. And how you do that is check emails regularly. A lot of things that happen, a lot of clubs, a lot of research opportunities, a lot of seminars are advertised through email. You should always be on top of your email. You should always know if something is sent to the students. You should always get it. So be sure that you're in the mailing list or whatever, however is it that you get emails. That's number one. A lot of the opportunities that I've found in my career has just been because I read emails and other people didn't. It's really that simple. Second thing is identify an advisor. As an undergraduate, you should have an kind of a core committee that will advise you on courses, on resources, and how to identify fellowships. This is very important. As undergraduates, try to apply to fellowships, try to apply to go to presentations like poster presentations, etc. That is a great way to highlight your work, and that is a great way to network and meet people, which in turn, it's like a cycle. So you go out to a seminar and meet people, and they kind of tell you, oh, do you know we have this fellowship coming up that you could apply to as an undergrad. So it's really networking, and I cannot stress this enough. You really have to know people. You really have to be aware of societies, like, oh, this is an undergraduate, like the ASBMB, which is like the American Society of Biochemistry and Molecular Biology. They have an undergraduate chapter. Enlist in their newsletter. They will send you, oh, this seminar is going on for undergraduates. This resource is there for undergraduates. So and never be afraid to ask questions, because that's how you really learn. If you're afraid to ask questions, you'll be in trouble. So go into lab. If you have a mentor, if you do research with somebody, they are an invaluable resource because more often than not, they'll be more experienced than you are. So they already have gone through this phase, and they can help you identify resources. So your undergraduate advisor, your research mentor, your counselor, all these people will help you identify resources, and also try to be part of clubs and stuff, because I find that when you are part of a club, you are like with people who are so motivated, and they always find the resources and they tell you about resources, and that's how you know resources. So it's like a kind of chain reaction. Try to get into things, and that's how you know about these resources.
If you have to do any bioinformatics or sequence analysis in your work, and also how did you learn how to analyze the RNA sequence for it?
Right. So in my line of work, so as a master's student, I did do some bioinformatics work, but I was really working with a faculty who specialized in bioinformatics. So he taught me a lot of the things that I know. So I have not actually ever sequenced RNA in my work. I more work with proteins that regulate these RNA. So it's more protein work for me. But where you can learn these resources is really, again, your lab. Your Someone in your lab will know how to do this, and they can teach you, and then you together you can kind of figure it out. So it's really, there's no book that you can read to get this information. It's really on job training. You make a lot of mistakes and you come back 2 steps, do it again. So it's like really on job.
That if they wanted to become a biotechnologist or a biomedical engineer, what paths would you recommend?
So you can become a biomedical engineer just with a bachelor's degree. You just have to go to college I think there are schools which give you a degree in biomedical engineering in 4 years. So that's a college degree, and you can do a great lot of things with that. You can even do research with that. But if you really want to be the leader, you want to be a group leader, you want to have your own lab in academia, or you want to be head of your own lab at, say, Merck or Moderna, something like that. If you really want to be on top of, towards the top of your field, you should get a PhD or an MD PhD. So you can get your 4-year bachelor's degree, and then you can apply to grad school or apply to med school and get an MD PhD, and you can be a biomedical engineer. So one of my cousins, she's a biomedical engineer, and so she went to Johns Hopkins, and her BS is in biomedical engineering. And then she went to UCLA and she got her PhD. And what she studies is she develops techniques that basically she will take your blood, and she develops technologies that detect cancer early in your blood. So the whole aim is to early diagnosis, so that the chances of the patient surviving the disease increases. So she kind of develops technologies for that. So she's a biomedical engineer, but she has 2 degrees, like a BS from Johns Hopkins, and then her PhD is from UCLA. So again, completely depends on what you want to do. You can even have a job with just my cousin could have had a job just with a college degree, but she wants to be kind of the group leader, like she wants to be on the so she went to graduate school. So you have both options. Depends on what you want.
If like what type of sciences are most used in infectious diseases, and what's the recommended route for that?
So if you want to be an infectious disease person, so you can again take the either you want to be an infectious disease expert like Dr. Anthony Fauci. He is an infectious disease expert. So you can take 2 routes. You can either be a scientist or you can be a physician scientist. If you want, for both, you have to get your first, go to college, get your again, it will be different for different colleges, but there will be some common core courses. You have to take courses in chemistry and biology and some math. And either you can be on the pre-med track, or you can, if you want to go to MD, PhD, or MD. So if you get an MD, you will be a doctor, and you can treat infectious disease patients. if you get an MD PhD, you can do both. You can do research and you can uh, treat patients. So when I worked with cervical cancer, my advisor, he was a physician scientist, which means that he's a surgeon. He actually operates on women who have cervical cancer. He also does research. So there are kind of two hats that he wears. And so he's a physician scientist. So he has an MD PhD. So as an infectious disease expert, you can be either just a doctor or both physician scientist and doctor, or completely different, you can be a scientist, like not the doctor part. Either ways, you have to go to college, get your bachelor's in chemistry, biology, biochemistry, whatever, and then get into either graduate school or medical school. That will be your track.
A lot of job opportunities, in your opinion, for biomedical engineering?
I would say so, and absolutely. The last, as you know, this centuries of biology, all our problems, even if you see a lot of Nobel prizes in chemistry, are all addressing biological problems. So we are biomedical engineers are at the forefront of technologies that we are developing to make our lives better, to help uh, cure diseases and help save the environment. So I think biomedical engineering is like it's a very in thing. I would say absolutely go for it.
We have our Instagram, at GirlsCodeIt. Feel free to follow us. And we also have our website where we will be posting things like the recording for this panel in case you wanted to, go back to some answers, or if you wanted to just watch the entire panel again for fun. You can find it on our website in the past events section. You can also subscribe to us on YouTube, Girls Code It, join our Discord server where we will be announcing more panels, more class opportunities, and just in general have a good environment for coding there. And that concludes today's panel.